Next presentation.
Okay, sir.
Sonali, you can stop sharing. Okay, thank you. Now, I invite Ms. Nazma Khatun to discuss phytoplankton diversity and its role in biomonitoring water quality of peat lakes. Please, madam.
Am I audible?
Yes, madam.
Hopefully visible too.
Your screen is not visible yet.
Okay, I'm sharing my screen. Hopefully the presentation is visible and also in slide mode.
Yes, madam.
Yes, it
Okay,
is very
thank
visible.
you. Good afternoon, everybody. Today I'm going to present my work on phytoplankton community, its ecological assessment, and how it can be used in water quality monitoring in peat lake ecosystem, and we are focused on Ranigaj coalfield. First, let me welcome you to the diverse world of phytoplankton. Whenever we think about aquatic diversity, generally beautiful colored fish, whales, and dolphins come to our mind. But beyond what our naked eye can see, there lies a diverse world of microscopic organisms. One such organism is phytoplankton. Phytoplankton are generally single cell photosynthetic organisms and they live suspended in the water. As they are capable of photosynthesis, they lead to the foundation of aquatic food web. They are the primary producer and they feed almost everyone in the aquatic system from microscopic zooplankton to multiton whales. And that's why phytoplankton is very important for any aquatic ecosystem. Now, the aquatic ecosystem we have focused on is the peat lake. Now, it is different from natural water body because it is created by open cast mining. And the main difference is it is generally bounded by high rock walls and there is absence of littoral zone. And if we compare peat lake with natural lake, for the same surface area, the depth is much more higher. As a result, the amount of water is also very high. Peat lake is also important because of its increasing number over time. And we have already said peat lake is created by open cast mining. And if we look at the 10 years data of coal production in India, it is almost doubled. Yes. And the contribution of open cast production also increased significantly. Mining leads to several problems from pollution to creation of barren land to creation of peat lake. And post mining, we need convenient and socially accepted solution. Maintaining peat lake as a sustainable aquatic ecosystem would be one such solution. Peat lake is also significant because of its huge water resource. We know that water is a valuable commodity, not at present time, but also in future it is going to be even more valuable. Local people already using this peat lake, and that's why it's important to understand the water quality, the aquatic diversity, and also we need to monitor the pollution status. And using indicator species, it can be done. Now we have selected phytoplankton as indicator species. Phytoplankton has already been used as indicator species for several studies and mainly because of its characteristics of being sensitive to the water quality change. It also has a very short lifespan and it gives us the real-time change in the water quality. And also phytoplankton can be easily studied under microscope. So after selecting our indicator species, we selected our study site. We have taken 7 peat lakes from Raniganj coalfield area. These are the geographical location of the peat lake. And we can see the location of the peat lake in the map of India, map of West Bengal, and Google Map. After selecting our study site, we have divided the whole study based on our 4 objectives. First objective was to analyze the taxonomic composition of the phytoplankton, then to study the phytoplankton diversity. We also wanted to analyze the seasonal variation of the phytoplankton. And last but not the least, we want to estimate the water quality of peat lake based on the phytoplankton diversity. Keeping in mind these 4 objectives, we have conducted our study for 3 years from 2021 to 2023, and we have visited the 7 sites and collected samples. And we have divided this whole year into 3 different seasons, pre-monsoon, monsoon, and post-monsoon. And for that, we went for sampling and we have used a very common method for phytoplankton sampling, and that is the towing method, and used phytoplankton net of 25 micrometer mesh size. We have filtered almost 70 liters of the water and collected the filtrate in a plastic bottle and fixed it with 4% formalin. We repeated the process for different seasons. And after taking the sample to the lab, we have went for Lackey's drop count method modified by Saxena in 1970, sorry, 87. We have taken the sample one drop per slide, and we have observed it under microscope, covering it with cover slip. We have used rose Bengal stain for better visualization when needed. And after that, we calculated the phytoplankton number per liter using this formula given by Leakey's drop count method, and it is modified by the Saxena, as I have already said. Now, after that, we identified each and every phytoplankton. We tried to identify up to species. Whenever it was not possible, we have identified up to genus. These are some of the standard literature that we have followed. And whole study can be summarized that we have found 66 species of phytoplankton belonging to 45 genus, 34 families, 26 order, 10 classes, and 7 phyla. We have excluded here the unknown species that we were not able to identify. So it was a good phytoplankton abundance that we have found in peat lake ecosystem. These are some of the phytoplankton, the image that I have captured. So we have found different species of amphora. We have found anabana in great abundance. We have found different species of cosmarium. We have found costium. We have found different species of Campbella. We have found the dinobrion in a very less abundance, but we have found it. We have found euglena. We have found different species of eunotia. We have found Gomphoneema vibrio in great abundance. We have found Merismopedia, microcystis in great abundance. We have found different species of Navicula. We have found Pilunaria, and we have found several species of Spirogyra. We have found beautiful Storastrum, and we have found some of the species that were not able to identify, and we put it in an unknown group. And we wanted to understand that this beautiful phytoplankton, what are the taxonomic representation of this phytoplankton? So first we try to understand the representation of different phylum. We have taken the representation both at the species level as well as the abundance level. At the species level, we see that Heterocontophyla is the most dominant phylum. It represents almost 40%. And when we look at the abundance level, again the same result, Heterocontophyla, yes, it was the most dominant phytoplankton phylum. But when we look at the second most dominant group, in case of species representation, Charophyta is the second most dominant group, representing 29%. But in case of abundance, we found the cyanobacteria was the second most dominant. As we can understand, the representation of the number of species belonging to cyanobacteria is less, but the abundance or the number of individual is quite high. Then we try to understand the distribution of classes of phytoplankton, and we have seen that the Bacillariophyceae is the most dominant class both at the species level representation as well as the individual level representation. But the second dominant group in case of species representation it was Zygmatophyceae, and in case of abundance it is Cyanophyceae. Next, we wanted to understand the diversity of this phytoplankton, and for that we have calculated different diversity index. We have calculated Margalef index, Maheny index, Hannon-Wiener index, Barger-Parker index, dominance index, Simpson index, and phyloevenness index. The formula is given in the slide, and we have calculated Maheny and Margalef to understand the richness, the Simpson and Hannon for diversity, Barger-Parker and dominance to understand the dominance of each and every species if present, and evenness to understand the evenness in the ecosystem. We not only wanted to study the different diversity index for each and every site, but also wanted to study the seasonal variation. So if we look at the abundance, we can see in most of the cases, in most of the sites, the abundance got reduced during the monsoon season, and it was highest during the post-monsoon season. When we look at the richness increasing during monsoon season, it is generally 45 if we take the mean value, and it was a minimum during the pre-monsoon season. The Mahenic index also shows similar result. It was highest during the monsoon season, and it is same for all. It has similar type of distribution for each and every site. Margalef index has the similar distribution, that is during monsoon it increases and for all the sites. Now the dominant index shows that there is no very seasonal variation and also the dominance level is less. Simpson index, which indicate the diversity, shows maximum result during the monsoon and the minimum result during the post monsoon, though there is a little variation depending on the site. Shannon index, which also represent the diversity, was maximum during the monsoon and minimum during the post monsoon. And again, we found there is a variation depending on the site. Barger-Parkin index, which also represent the dominance, we see that there is not very seasonal variation in case of different site. And Pylos evenness index, indicating the evenness, shows a high level of evenness in each and every site, and it is generally maximum during the pre-monsoon season. Last, we wanted to understand how this phytoplankton can be used to understand the pollution status of peat lake ecosystem. Now, one index, that is the Shannon-Wiener index, is used to understand the pollution status, and different scientists have used it. Two scientists, Staub et al. In 1970 and William and Doris in 1968, had given a relationship between Shannon-Wiener diversity index and the pollution status. As we can see in the table, there is a range of Shannon-Wiener diversity index and the pollution status it indicates. Using this information, we wanted to understand how is the pollution status of our peat lakes and how it varies with season and whether there is a pattern or not. So our result shows two interesting patterns. First, the water quality or the pollution status of pre-monsoon and post-monsoon was moderately polluted if we take the classification based on William and Doris in 1968. And the water quality get better during the monsoon season. There is an exception in JBP1, one of our site, but the water quality comparatively gets better during the monsoon season. Another interesting fact was that one of our site, that is GEP, showed a clean condition or slight pollution throughout the season. It indicates that the water quality of GEP is quite better if we compare it with other sites. GEP becoming the oldest peat lake might be one reason for it, but we need further study to confirm it. Another index that can be used to understand the pollution status is the Margalef index. Now, as we can see in the table, this is the range of the Margalef index and this is the water quality it indicates. The Margalef index pollution status also showed similar result, though there was a little variation. So in case of pre-monsoon and monsoon, the water quality was moderately polluted in most of the site, and the water quality got a little bit better during the monsoon season. So from all this study, we can conclude that the phytoplankton is in abundance in peat lake ecosystem, which indicated the sustainable aquatic food web, which is a very good sign if we want to use peat lakes for sustainable aquatic ecosystem. Now, the most abundant phylum we have found it Heterocontophyla, and the most abundant class was Bacillariophysi. We have found phytoplankton diversity with low dominance, and the abundance was minimum during the monsoon season. Also, there was seasonal variation of different indices. Mehnick, Margalef, Simpson, and Shannon all were highest during the monsoon season. We have found the dominance index and abundance was highest during the post-monsoon season, and the highest evenness we have found prior to the monsoon season. And also the pollution study can conclude that during pre-monsoon and post-monsoon, the water quality was moderately polluted and the water quality got better during the monsoon. Overall, if we take the mean value, then the water quality of the peat lake was moderately polluted. These are the references that I have used. Thank you for your kind attention, and I'm open for questions.
Thank you, madam. Questions, please.
Yes, sir. I have a question again. Can I go ahead?
Definitely, yes. You can go ahead.
So, ma'am, my question was you have used various indices for diversity. Uh, can you explain that why many indices were used, like richness? Margalev richness was also used, Shannon Wiener was also used. Why so many indices?
Okay. So yes, first we wanted to understand the diversity from multi-dimensional level. So we have taken Mehnik and Margalev in order to see the richness, and we wanted to also include the evenness, and that's why we have taken the Shannon and the Simpson index. And again, we just wanted to see the evenness, and that's why we have taken Pylos evenness index. Now, yes, why two index for the richness and why two indices for the evenness? Two indices for the diversity. Mainly because we have different sites. So, and each and every site has slight variation. And we wanted to understand the diversity from multi-dimensional level. And that's why we kept all the indices in order to understand. I have not gone into the details how this diversity is represented in different sites. Rather, just only I have focused on the seasonal variation. So we wanted to understand the diversity for each and every site in great details, and that's why we have kept all the indices.
Okay, ma'am. And another continuation question,
Yes,
like the diversity in the monsoon season decreased, right?
yes,
According
yes,
to your study,
yes.
uh, the
No, no,
decrease.
no. The diversity increased during monsoon. The
Sorry,
diversity,
the abundance I think decreased, right?
abundance decreased. Yes.
The the decreased abundance can be, you know, the dilution factor because of rains, maybe.
Yeah, it might be. We are also assuming it to be the reason because of the rainfall, the amount of water increase, and because of the dilution factor, it might be a reason that the abundance got reduced.
Okay, ma'am. Thank you.
Okay, thank you so much. And I can also see one compliment. Uh, nice presentation. So thank you, Raghavendra. I hope I'm taking the name in the right way. And Nivedita also complimented. Thank you. And one question is there. With the due permission of Topojit Babu, Topojit sir, can I take the question?
Yes,
Oh
yes.
no, no. Oh, this is the previous question.
No, it is not for you. It is
Uh,
not for you.
it was for Sondipa. Okay,
It is
okay, I got
personal.
it. Thank you.
Thank you, madam. Thank you so much for this nice presentation. So we move on to the next presentation, again on plankton from